Hello. In this very short presentation, I'm going to talk about how the neural connectivity of the brain is going to change depending on the state of the brain. Either you are looking at the resting state, functional connectivity of the cervix as a normal, or when a psychosis like a schizophrenia happens, how the functional neural connectivity of the brain changes. You can find both the data sets, the slides, and also a report, which is a summary of all the steps which has been taken from the data manipulation to data processing, and also the method on the GitHub, which is listed at the bottom of the window on the screen. Now, as you can see in this adopted slide, you can see that we have three statistical spatial correlations between different parts of the brain as a activated parts of the brain. You can see that, for example, the green one is an external attention network, which the dots at the bottom reflects a graph of the resting state connectivity, the control network, and also you can see at the bottom the psychosis at the default network of the patients at the resting state. The functional MRI has been obtained. Now, you can just look at also at the report at the GitHub, but also the data as it has been shown, it has been just used as for downstream analysis, has been taken from this kind of a setup that we just localized structural based morphology of the brain and functional neural connectivity of the brain, and also we just did some kind of algorithms to find the graph of the connectivity of different spatially co-activated part of the brain during the resting state, which means that the subject under the fMRI scan has nothing to do except daydreaming and in the absence of the external stimuli. Now, I'm just going to go back to the data sets and the codes which has been prepared. The hypothesis for this project has been that we assume that when we just obtain the data from the resting state, functional MRI imaging of two different populations. The first populations, they're just going to be healthy, normal cohorts, which are not diagnosed with any psychosis. And the second cohort are the patients which they have been clinically diagnosed as those who show symptoms of schizophrenia. Now, our hypothesis is that the resting functional MRI, which represents co-activation of different parts of the brain, which we just call that functional neural connectivity, within these two cohorts is different. Then our null hypothesis, it's going to be like that. We say that, okay, two different cohorts, the resting state, functional connectivity is not different from the resting state functional connectivity of the schizophrenic patients. There is no difference whatsoever. And the alternative hypothesis, which we are just going to use that the graph theory in order to show that, we say that, okay, the functional connectivity between these two cohorts of these two kind of normal and schizophrenic patients are going to be different. Now, as you're going to see that, as you're going to find it on the GitHub, at the first step, I have installed the dependencies because for this analysis, I'm just going to use a deep autoencoders, then decode the latent representations of the inputs, which they are the features and the functional recording pre-processed from the fMRI scans from both cohorts, either schizophrenic and normal. Then from the latent space representation, training the model in order to produce the graph which shows the connectivity of different recordings which has been obtained from the resting state fMRI scans. Now, as you can see, as a first step, I just pre-process the data. For the data pre-processing, I just divided the datasets into two different kinds. The datasets, which represents the functional neural connectivity of the healthy and the functional neural connectivity of the schizophrenic patients. And as you can see, the datasets from these recordings show 380 features. And you can we are going to exclude also the ID from those every individuals that we had. Then as a result, the total recordings for each subject is 379 recordings, which each recordings represents the functional connectivity of that subject. Also, I just divided the datasets for healthy and schizophrenic patients. And as a very most important steps before I run it into the deep autoencoders, which is written in keras, I made sure that I have normalized the recordings which I obtained from the experiment. These are just the pre-processing steps as step number 1. You're going to see that step number 2, I define the graph structures for the healthy populations. I provided, for example, as you can see, I said that, okay, I just dropped the class and also the ID. Then you're going to see that I just started for each subject with the recording, which totaled to 378 recordings, which each recording represented a measurement of the functional connectivity of the brain as a statistical correlations that we just pre-process and find it. We just define the model. For the first one, I just used the healthy samples. And as you can see on the diagram which is above, I showed that from the picture which is taken from, I should say, from the keras websites to represent a conventional autoencoders, which you can see, for example, number 2 on the left side, which is which shows that, for example, the inputs that we have, the inputs is gonna be taken into the encoder part of the system. Then it compresses the representation. Then it produces the latent space, a latent representation of the data sets that we just started with. Then if we can start with the features that they are not very relevant as an input data. When you encode it into the latent space, you only extract the most relevant features of that particular subject. Then we just stop there. And we then started to find the graph from that latent space before we decode it to extract the noises. And that's going to be the methodology which I used for this experiment. Now, speaking of which, I first started with the model A. With the model A, I just defined the decoder and the encoder. And I trained the model. And I just ran 50 epochs. And I choose the batch sizes to be 32, and I just train the model and fit it on the healthy patients, which I normalized that before. Then at the end, when the training was done, I just extracted the embeddings, which it means that I use the latent space representation of the data. Then I just used, for example, a first type of the clustering, unsupervised clustering. Then you can see that the first clusterings resulted in 46 clusters. It means that the recording which I had, when I removed all the unnecessary and noisy measurements of each subject, it can be considered at 46 clusters of the data. It can represent the 46 pairwise nodes or 46 parts of the brain at that particular time that they were co-activated during the resting state of the healthy patients. And that's a two-dimensional diagram representation of the clusters. Then at the end, I just show the plots, show the graph of the connectivity of different nodes. Then you can see that on the first model, which the hyperparameters, for example, was MSE, for example, for the loss functions, on the 50 epochs, the connectivity of the healthy patients is this. But in the second part of the code, as you can see, I changed the hyperparameters of the model. For example, for the hyperparameters of the model, I choose a different optimizers, and I choose a different learning rates. And you can see that when that happens, and when I train the model, and you can see the plots of the loss function of the second model, which I use autoencoders to extract the embeddings from the latent space, you can see the loss function which I choose optimally, it just decreases during that 50 epochs. And still, in the second model, when I change the hyperparameters, I got 46 clusters as I got before, which they can inherently explain that, for example, at that particular time of the resting states, the sample of the subject of the normal people, they had 46 collected nodes that they were co-activated. And you can see the graph, the surprising thing was that when we choose different hyperparameters, the connectivity of the functional networks was different, which it showed the sensitivity of the model from the latent space with respect to the connectivity of different parts of the brain. Now, for the second parts of the codes that you're going to see, I just choose the datasets from the schizophrenic patients. You can see that I have 3 different layers. I have input layers, I have encoder layers, which I just choose the deep autoencoders. The first one comes with 60 nodes. I just regularize that in order to avoid the over learning by dropout method. And I just extracted latent space and I just put that into the decoder layer for different neurons, 32 neurons and 64 neurons. And you see the bottleneck which I have at the latent space that selectively represents and extracts the data which is most relevant. I just run the 50 epochs, and as you can see, still, with the schizophrenic patients, I showed 46 clusters, which it can imply that with the schizophrenic patients, still like the healthy patients, the coactivity of different parts of the brain, it shows 46 clusters. Each part that they are activated, they come under one cluster. And this is the 2-dimensional representation of the codes. And the graph of the connectivity, it shows something like that. Which if you go back and to compare that with the functional connectivity graph of the healthy patients, you see that it's a widely different pattern of the connectivity between different nodes that is taken separately from both the healthy and the schizophrenic patients. Now, because I did the same thing for the healthy patients, I just choose different hyperparameters of the deep autoencoders for the schizophrenic patients, which I call that model B. You can see the code is represented there. The optimizer is different. The learning rate becomes different. And by the changes of the hyperparameters, you see the loss functions after 50 epochs drastically drops. Still, it shows 46 clusters of the co-activated part of the functional neural connectivity of the schizophrenic patients. But you see that, again, it shows sensitivity to the hyperparameters when it shows that to the clusters. Now, there are many other things, and also at the last part, you're going to see that I just choose other methods, which is a Bayesian representation of this pattern of the connectivity, which is called variational autoencoders. Now, then with this one, as you can see, in these projects, what we expected to do, we tried to show that from the data that we collected from the resting state functional MRI, we tried to show that the pattern of the connectivity is going to be different between the healthy subjects and the subject which is diagnosed with psychosis and schizophrenia. The dataset and the paper can be found at the link below. Thank you.